Okay, I think we're gonna get started because we have a different format today. We have two speakers from MIG. But first, I'd like to acknowledge the traditional custodians of the lands and the waterways where we meet today on the Parkville campus, and pay my respect to their elders past, present, and emerging. So good afternoon, everybody. I'm Kima Neka. I'm the director of Melbourne Integrative Genomics. And it's my pleasure to kick off the seminar series for this year from Melbourne Melbourne Integrative Genomics with our two postdocs from MIG. So we have Jiadong Mao, who's going to start. So Jiadong has a PhD in statistics from the University of Melbourne. And he's been a research fellow in my lab. In the past 2 years, he's interested in advancing biomedical research, especially in stem cell and cancer biology, to provide new insights into complex cellular processes. So let me thank Jiadong for presenting today and invite him to the floor.
Thank you, Kiman, and welcome to our first MIG seminar this year. So I'm very glad to talk about my work on spatial transcriptomics. So the topic today is a computational package we developed in the last 2 years. So the first generation is called PhiSpace, and the first generation of PhiSpace was developed for single-cell multiomics data. And this is the second generation, PhiSpace ST, and it's developed for spatial transcriptomics. So I've been using the title A Tale of Two Spaces for some time now. And I think it's a very good title because it summarizes what I do in this project very well. So the subtitle is Discovery of Spatial Biology. So that's the first space. In phenotype space. So that's the second space. So that is to say, we analyze spatial transcriptomics data, but not in the original RNA gene expression space, but in the so-called phenotype space, which is a more appropriate space for describing the complex cell states that you see in this type of data. A bit of background, first cell type. So ever since the invention of a microscope, scientists have been trying to describe and document different types of cells, but mostly based on morphology, that is the shape of the cell, function, what they do in physiological processes, and their location. But with the advancement of high-throughput sequencing technology, especially single-cell and spatial technologies, now, in addition to those phenotypes, we can actually dig deeper into the molecular level. So we can look at different cells and measure their gene expression levels, protein expression levels, chromatin accessibility, and histone modification. So we know much more in much detail about what cells do. And so, and I think by far the most popular type of high-throughput sequencing data for describing or annotate cell types is single-cell RNA-seq. And so in this type of data, we simply group cells with similar transcriptional profiles into clusters. And then we find the marker genes and try to give them different names to describe them. So what we do nowadays, because we have so much data and we can't afford to annotate every cell by hand, so reference-based methods have been developed, data-driven methods. So these methods basically is based on classification algorithms. So the idea is you need to have a well-curated reference data set with a lot of cells, including rare cells, and then you annotate them using cell type labels. And then when you have a new unannotated query data set coming in, you train a classifier. And then, so a classifier takes transcriptional profiles as input and cell type labels as output. So you apply that classifier to unannotated query cells, and then you derive some cell type labels. Now that's what people typically do. But actually, I would say that actually cell type classification does far more than that, because it's not just gives you black and white annotation, cell type labels. It can only do uncertainty quantification, which means if you imagine you have a bunch of cells which have a novel cell state, and when you apply a good classification algorithm or reference mapping, then your algorithm will tell you that these cells do not look like any existing cell types in the reference. And that's usually a very good indicator to tell you that it might be a new cell state. So cell type classification is also good for working. It's also good for the discovery of a new cell state. New cell state. Yes. So spatial transcriptomics. So in addition to high throughput sequencing measurements of RNA, spatial transcriptomics also tells you the spatial location, spatial information about those RNA molecules. So what it does, there are many different kinds of technology, but in general, what they do is they try to retain the spatial organization of the tissue organization, spatial organization of cells, and then they do sequencing. So if single-cell RNA-seq tells you which cells are doing what, then spatial transcriptomics tells you which cells are doing what and where they are doing it. And when you analyze spatial transcriptomics data, obviously cell type annotation is still a key step. But well, this is, at least in my experience, when you try to get your hands dirty in this field, especially when you have a single cell background, the first problem you notice is that there is no cell, or at least no single cell, in spatial transcriptomics data. Because with all these fast evolving technologies, some of them give you super-cellular resolution, such as Visium, which means they can measure what's happening in a big spot. So they measure all the RNA molecules in a big spot. But that spot contains multiple cells. On the other hand, you have sub-cellular resolution. So these platforms are often single molecule resolution, which means you can measure individual RNA molecules and you know their spatial coordinates, very, very high resolution. But again, you don't have single cells. And between them, you have near cellular resolution. The reason why you don't have single cell is because there is no, well, physically, there's no cell isolation happening. Because as I said, in order to retain the tissue organization, the organization of cells, you cannot afford to dissolve the tissue into single cells as you do in single cell technologies. So actually, you do not have real single cells. So a common problem for this data type is you have to deal with a mixture of cell types or cell identities. That is because it's obvious for multicellular or supercellular resolution, the best you can get are those spots with multiple cells in them. But even if you try to apply some deep learning imaging-based cell segmentation to subcellular technologies, that is, you try to figure out the cell boundaries in the tissue, because they are not real single cells and they might not be that accurate. So each segmented cell may still contain transcripts from multiple cells. So you constantly have to deal with the problem of mixing cell identities. So an analytic tool you have for that type of problem is called cell type deconvolution. So in a conventional cell type deconvolution algorithm, you use single cell RNA-seq to decompose each spot, say Visium spot, into cell type proportions. So you may have, say, one spot containing 70% of CD4T, 20% of CD8T, and 10% of natural killer. And those proportions add up to 1. So the goal of this type of algorithms is to understand spatial distribution of known cell types. But what we do in 5-space ST is that we still rely on single-cell RNA-seq as reference, but we really try to deconvolute every cell-like object. So we're not restricted to multicellular spots, but also segmented cells and spatial bins. Spatial bins means you simply bin or tile the tissue space into smaller bins, and you try to annotate those bins. Because that is because we noticed that the problem of mixing cell types exists in all these cell type objects. So, and another innovation is that we do not restrict the cell type scores to add up to 1. Now that is actually motivated by cancer research, because if you try to look at a cancerous tissue, what you will notice is that the tumor region is far more active, transcriptionally active, than a non-tumor region. So if you apply file space, ST, to annotate those cell-like objects in the tumor region, you might get things like my cell-like object or that region is 80% similar to epithelial cell, 50% similar to stem cell, and 20% similar to immune cell, and they add up to 1.5. That's because that the epithelial identity reflects the origin of the cancer, 50% stemness, and that 20% probably reflect immune infiltration. So, but if you look at a real, a non-cancerous region, a normal stromal region, what you might get is 80% epithelial identities and 20% immune identities, and they add up to 1. So we do not restrict the proportions to add up to 1, so that you can, so this leads to a very natural measurement of cell state divergence. So you can actually measure which region have more divergent cell types compared to other regions by using phi space. So a bit of history of phi space, as I said, we first applied it to single cell multiomics, and this is the second generation, and we have a very interesting divergent team. So some comparisons, yeah. So after we developed the first generation of phi space, we tried to work on spatial transcriptomics. And in addition to the lack of single cell resolution problem, the other thing is we noticed that there are some limitations of existing deconvolution methods. Most of them are specialized in specific types of cell-like objects. If you read those papers, typically they were developed for platforms like Visium, where you have multicellular spots. And they often rely on a single reference dataset. They cannot easily handle multiple reference datasets. And they are specialized in well-defined cell types, and they don't really talk about how do you describe new cell types or new cell states, rather. A lot of these methods are really, really slow, computationally, lack computational efficiency. And the last problem is more statistical, but it also has practical consequences, is that a lot of these methods rely on restrictive parametric assumptions. And I have to highlight variational autoencoders because it's a very popular type of models. But variational autoencoders are a type of Bayesian model, which means you have to assume the probabilistic distribution of the data generating process. So when you try to apply a variational autoencoder, VAE, developed for transcriptomics to attack-seq, you can't do that directly because the data distribution changes. So you have to tweak the model to make it work for a new type of distribution. That's probably why it's so popular, because every time you change a omics type, you need to develop a new model and hence a new publication. But from a user's perspective, it's not good because that means this type of model is not that flexible for new emerging data types. And phi space overcomes all those problems, and it's very fast to compute, and it's non-parametric. That's why we could easily extend a method developed for single-cell multiomics to spatial transcriptomics. How does it work? So very briefly, first, for input, you can take several single-cell reference datasets with cell type labels. The reason why you want to use more than one reference is sometimes you simply couldn't find a single dataset with all the cell types you want. So sometimes you need multiple. And on the other hand, you have those RNA expression levels for cell-like objects and their spatial coordinates. That's your query data. And then the model is very simple. It's POS. So that's why it integrates very naturally into the MIG Seminar framework, which is also based on POS. So the idea is you train a PLS regression model or soft classification model using each reference, and you apply those PLS regression models to your query. And then in the end, you simply stack or concatenate all those annotations, and you get what we call phenotype space embeddings. So phenotype space embeddings of cell-like objects are simply a matrix like this, a continuous matrix. Each column is a cell-like object, and each row is a cell type. So you are using the cell types defined in all these references to try to describe or characterize the cell state in your query dataset. And because we do implicit rescaling of these annotations, so that means these annotations are actually very comparable across different reference datasets and also across query samples. So if you use the same reference to annotate multiple query data, there's no batch effect. So you can directly use the phenotype space embeddings as the input for multi-sample downstream analysis. So there are two types of analysis we look at in our manuscript. The first type is you can use these phenotype space embeddings, and you can do a simple clustering. And you can get some spatial niches. For example, here I show you tumor region, tumor niche, immune niche, and stromal niche. And then as what you do in differentially expressed gene analysis, you can do a enriched or differentially expressed cell type analysis. So you can find the differentially enriched cell types in each of these spatial niches. The other type of analysis you can do is for each sample, you can derive what we call a cell type co-presence matrix like this. So this co-presence matrix tells you which cell types tend to be co-present or co-located in the same spatial region. So it's like it's almost like a correlation matrix. So you can summarize the co-presence, cell type co-presence or co-localization of each tissue into a matrix like this. And then if you plug this into a discriminant analysis algorithm, you can figure out the co-presence patterns of tumor region or non-tumor region. So that is to say, you may find some cell types tend to be co-present more in tumor region compared to non-tumor region. Now, the first example I'm going to show is a Visium dataset. So again, it's developed by 10x, and the cell life object in that platform is multicellular spot. And what's special about this dataset is that it contains 18 healthy and cancerous lung samples from non-small cell lung cancer patients. And the reference here, I'm not using multiple reference because this is a benchmark study. I need to compare PhySpace to other methods, so they couldn't handle multiple reference. So here I'm just using an integrated human lung cell atlas. It's part of the Human Cell Atlas project. And then the challenges here is, of course, you need to do interpretable cell type deconvolution, and we are using a healthy reference to annotate a cancerous query samples. So how do we interpret that? Now, that is one example, one tissue sample from this data set. I plotted some marker genes here. As you can see, it consists of these rather large spots. And the reference contains 61 healthy cell types. Now, if you look at the visualize the annotation results as a spatial heat map like this, so I'm comparing PhySpace to these three state-of-art methods. They are really widely used. The first thing you notice that, so if you ask them to predict B cell abundance, so you want to know the spatial location of B cells, or the rather B cell identities, you get really different answers from these methods. So which one should I trust? I was really shocked by this because I expected these methods to give you more or less same results, but actually no, very different results. So that motivated us to do a benchmark study but how do you benchmark? Because you don't know the ground truth. You don't know the true cell type proportions in each spot. So we used a proxy of truth, which is based on a very simple assumption. The idea is if you say a spot has high B cell identity, then that spot should at least express B cell marker genes. Well, it is a very reasonable assumption. And then based on that, you can define meaningful markers. So we define meaningful markers as the intersection between these two gene sets. So the first gene set is the spatial differentially expressed genes in high B cell abundance area. So if a method predicts an area to have high B cell identity, I try to find the DE genes in that area. And then, because on the other hand, I know the cell type marker genes because I have a reference data set. So I look at how much these two gene sets overlap. And for B cell in that particular tissue, we found more meaningful markers from a phi-space result. But that's just one cell type and one tissue. You can do that for all 61 cell types and all tissues. What you find is that phi-space and RCTD, so these two methods tended to give you higher number of meaningful or interpretable markers. But RCTD was really very, very slow. So TACO, on the other hand, was very fast, but it gave you rather low number of meaningful markers. So that gave us a lot of confidence because it means phi-space is applicable and it's meaningful and fast for processing spatial transcriptomics data. Now, the other powerful feature of phi-space is that you can do a multi-sample analysis like this. So remember, I said we have 18 tissues. So here I calculate one cell type co-presence matrix from each of these tissues. And then I do a very simple PC analysis of these matrices. So PC1 and PC2. And what you see is that they group very nicely into these clusters according to conditions, according to disease types. So healthy and background. Background means non-cancerous tissues from cancerous patients. So healthy and background stay close together, and they tend to have negative PC1 score. On the other hand, all these cancerous tissues tend to have positive PC1 score, and they were grouped into cancer subtypes. So lung squamous cell carcinoma, LUSC, are here, and adenocarcinoma are here. And when I looked at this graph, I thought, because we know that this type of lung cancer tended to have, this is the red one, tend to have worse prognosis than the purple one. So does it mean PC1 represents some kind of prognosis? But sadly, we don't know the true prognosis outcomes of those patients. So we use the data-driven approach to calculate the favorite prognosis scores of all those samples. And what you find is that this patient 24, it's a adenocarcinoma patient, but it had a really low favorable prognosis score, which means it had a very bad prognosis score. So that's probably why it tended to be located closer to squamous cell carcinoma patients. So you can actually interpret the relative location of these patients, and you can attribute that to prognosis, possible predicted prognosis. Now, I'll quickly go through this example because it's biologically more interesting. This is from Mark Dawson's lab from Peter Mac. And here we're looking at sub-cellular platform, scRNA-seq. And in addition to having a mouse spleen with acute myeloid leukemia cells, all those AML cells were actually contain barcodes. So because people in the lab, they applied splinter linear tracing, a technology developed by them, so that each AML clone cancer clone in this spleen will have the same barcode. So you actually know where all those cancer clones are located. And here we release the full power of phi-space by using 4 references because we couldn't find a single integrated, well annotated mouse spleen atlas. So that's why we used 4 references. And then what you notice, I should remind you that this is leukemia, so it's liquid tumor. And the poor mouse was at the last stage of their life. So the cancer cells were really everywhere. And it's, but still, you can see some spatial patterns of these individual clones. Some don't, like this one, it doesn't, but some do have some spatial structures. So this is a less well-documented phenomenon because we don't really know how a liquid tumor tend to be spatially distributed. So we thought of a method to, because there are thousands of clones, so we thought of a method to summarize the typical spatial locations of these clones by defining these so-called meta-clones. So the idea is you cluster those spatial beams according to their compositions of clones. So that is to say, this pink cluster meta-clone tends to, so all the bins within this clone tends to have the same individual cancer clones. And you ended up having these meta-clones. And of course, on the other hand, you have 5-space annotation. So actually you find that some clones are more neutrophil-like, some are more lymphocyte-like, and some are erythrocyte-like. So they probably, do they reflect different origins of cancer, of leukemia? Or do they exhibit different, do they have different resistance to different therapies? That's the next step, because here we only have one sample. Okay, so a summary, a quick summary. So 5-space ST goes beyond single sample, so we can do multi-sample analysis very fast. And then this is more like a philosophical question. That is, cell segmentation is not always needed. It really depends on biological question, because if you just want to know the overall pattern of a certain identity or certain cell states, then you actually do not need to have very accurate cell segmentation. And cell segmentation is often very time consuming and somewhat inaccurate for some tissue types. The last thing is we have successfully applied 5-space to bulk, single-cell RNA-seq, single-cell multiomics, and right now to spatial transcriptomics. And I can see no problem extending it to spatial multiomics or even finding developmental trajectories in space. So that's it. And the manuscript has just been put on bioarchive, and scan the barcode and have a read, because I didn't talk about all the case studies. Yeah.
Happy to take one very quick question. I'm just, conscious of time, and I want also to leave time for Roshan. Is there one question from the floor to Jiadong? Yep. Eva.
Hi, thanks. Really nice talk. I was wondering, what would the output of 5-space be if you have a cell type in your spatial data which you don't have in your single cell reference?
Oh yeah, that's actually we have that in every case study, because you have cancer cells in the query, and you do not really have cancer cells in the reference. So what you get is you can look at so actually all these are cancer cell states. So they will tell you some cancer cells are more like neutrophils, granulocytes, and some cancer cells are more like lymphocytes. Yeah.
So they become like divergent, like a mix.
Yeah. And they all have rather high scores because they are it's more active, the cancer cells. Yeah.
Okay. Thank you, Jiadong. I think we're going to move to the next speaker. We have an issue. Microsoft is trying to update at the same time, so I don't know what's going to happen.